Hello, my name is Ryan Morais, and I'd like to share my project focused on lichens, which are a symbiotic association between a lichenized fungus, or a mycobiont, and an alga, or cyanobacterium, or a photobiont. More specifically, this study focused on the lichenized fungus Firmicillin cephalota and its green algal symbiont Trebouxia. The focus of this project was to use analysis of DNA sequence data to characterize the diversity of mycobiont and photobionts found in the morphologically defined lichen Vermicillin cephalota, and to use this understanding of diversity to test the specificity of the two symbionts. Vermicillin is a fruticose lichenized fungus endemic to coastal regions along western North America experiencing regular fog. Specificity refers to the range of a species a given symbiont will associate with. The degree of specificity between two symbionts varies, ranging from generality, where one microbiont can associate with multiple photobionts, to specificity with a one-on-one species association. Mode of reproduction also may play a role in specificity, as lichens can reproduce sexually, dispersing without the photobiont, and asexually, which couples with the photobiont. Samples of lichen were taken from the west coast of the United States of America, as seen in the diagram to the right. My three hypotheses for the study are as follows. First, the lichenized fungus of Vermicillin cephalota S. lat is comprised of multiple cryptic species. Secondly, multiple cryptic lineages of algae associate with these lichenized fungi. This is based off a study by Spajut et al. 2020, revealing that there are cryptic lineages of V. cephalota in Mexico. And lastly, symbiotic pairing is strongly taxonomically related, or species-specific, based on reproductive co-dispersal for V. cephalota as compared to other representatives of the genus. After DNA was extracted and sequenced from both the mycobiont and photobiont of the samples, species were phylogenetically compared to earlier delimitation studies by Mugia et al. 2020 and Spajut et al. 2020. Two phylogenetic trees were constructed, one for each symbiont. For the mycobiont tree, at least two molecular species of lichenized fungi are currently circumscribed under the name of Vermicillin cephalota. These two species appear to be allopatrically distributed, with one species found throughout the entire west coast of the United States of America. The second species of Vermicillin cephalota was exclusively found in the Baja region of Mexico. More sampling is needed to determine whether both V. cephalota species can be found within the Baja region, and the phylogenetic tree of the photobiont shows that the algae in the tree are not a monophyletic and suggests that one species of Trebouxia associates with V. cephalota, meaning that incredibly, over the 1,000 miles from the upper end of Washington state to the lower end of California, the photobiont does not change. A bipartite network was created to visually display patterns of specificity. Gray lines between the upper green photobiont and lower purple mycobiont boxes represent an association between these two symbionts. This diagram emphasizes the high specificity of V. cephalota as it only associates with a single species of Trebouxia. Of the five Vermicillin species, V. cephalota and V. zebrina live on bark, while V. comboides, V. levigata, and V. procara are rock-dwelling species. These sampled bark lichens predominantly reproduce asexually and display a much higher degree of specificity as compared to the more generalist rock-inhabiting fungal species which reproduce sexually to a much greater extent. This provides evidence that the degree of specificity is influenced by the primary mode of reproduction, as lichenized fungi that disperses with its algal symbiont are more likely to be highly specific with that symbiont. The implications of this study are a better understanding of species boundaries in these lichens, providing taxonomic resolution and further increasing our understanding of evolutionary relationships, and further evidence for asexuality linked to generality. Taxonomic understanding gives us a framework for conservation and public policy, as we cannot make informed decisions without first understanding what species we are working with. This study will be a small part of the continuous endeavor for taxonomic resolution of these fascinating organisms. Thank you to the UConn Office of Undergrad Research for funding this project, and thank you for your time.